Hello there, and welcome to Sleepy Science Vault. The human mind holds something strange within it, a quiet paradox that most of us never stop to notice. We spend our lives collecting moments, conversations, faces, mistakes, small victories, trusting that our memories will simply stay where we left them. And yet, so often, they don't. A name slips away mid-sentence. A childhood afternoon disappears entirely, while a single smell from that same year can suddenly bring it rushing back, vivid and whole, as if no time had passed at all. For centuries, people assumed that memory loss was simple. That forgetting was just fading, a candle slowly burning down until the light was gone. But the more scientists have studied the brain, the more that simple idea has quietly fallen apart. What has emerged instead is something far more intricate, a picture of memory not as a fragile candle, but as a living, shifting system, constantly making decisions about what to keep, what to release, and what to reshape. Tonight, we are going to explore that system slowly, one discovery at a time. Not to alarm you, and not to overwhelm you, but simply to walk gently through what modern neuroscience has learned about how memory works, and how it sometimes doesn't. There is no need to follow every detail closely. You can simply listen, and let your mind drift as the science unfolds around you. Let's begin with an idea that quietly reshapes how we think about forgetting. Memory loss, in many cases, is not actually about losing information. It is about losing the path back to it. This distinction feels small at first, but it changes everything. For a long time, the assumption was that when something is forgotten, it is because it is no longer there, it was erased, or it decayed, or it simply vanished from the brain's storage. But researchers have found that in many cases, the memory itself may still be sitting quietly inside the brain, fully intact. It is the retrieval that has failed, not the storage. Think of it like a vast library, with millions of books placed on shelves that stretch farther than the eye can see. Sometimes, a book truly is missing. It was never properly placed on the shelf, or it was accidentally destroyed. But other times, the book is exactly where it always was. It is the map to find it that has gone missing. The catalog card is lost, or the shelf number was never written down clearly enough to search for later. This is why a memory can suddenly return, seemingly out of nowhere, triggered by a smell, a song, a place, or an old photograph. The information wasn't gone. The doorway to it had simply been closed, and something, quite by accident, opened it again. This single idea has quietly shifted the entire field of memory science. It moved researchers away from asking only why did this fade, and toward a more curious question. Why can we no longer reach it? And that question leads naturally into our second idea, one that may feel even more surprising. For much of the 20th century, forgetting was treated as something passive. As though memory simply eroded over time, the way stone erodes under wind and rain, slowly, quietly, with no real mechanism driving it forward. It felt like decay was simply the natural fate of information, and the brain was a passive victim of that decay. But many neuroscientists today understand forgetting very differently. They now see it, at least in part, as an active process. Something the brain actually does, rather than something that simply happens to it. This means that in certain cases, forgetting is not a failure of the system, it may actually be the system working exactly as intended. The brain appears to have mechanisms that intentionally weaken or suppress memories, particularly ones that are no longer useful, or ones that might interfere with newer, more relevant information. It's a strange thought to sit with, that losing a memory might, in some circumstances, be less like a broken machine, and more like a garden being tended. Some growth is encouraged. Other growth is quietly pruned back, to keep the whole space healthy and functional. This idea connects closely to something researchers now describe as viewing memory loss through the lens of control, rather than simple failure. In this view, the brain is not just a container that holds memories until they either survive or disappear. Instead, it behaves more like a constant balancing act, always managing three competing needs at once. It must remember what matters. It must suppress what would only create confusion or clutter. And it must reorganize older information as new experiences continue to arrive. Imagine, for a moment, an enormous control room, filled with countless dials and switches, adjusting subtly, moment by moment, day by day. Nothing stays fixed for long. Some signals are turned up. Others are quietly turned down. The brain, in this sense, isn't simply storing your life, it is actively curating it, deciding, again and again, what deserves to stay close at hand, and what can safely fade into the background. This is a very different picture from the old idea of memory as a warehouse, where things simply pile up until they are lost under dust. It suggests something almost intentional, a kind of quiet intelligence behind the process of forgetting itself. And if the brain is managing memory this carefully, it raises a natural question. Where, exactly, does all of this happen? For a long time, people imagined there might be a single place in the brain, a sort of vault, or memory center, where everything was neatly filed away in one location. But this turns out not to be true at all. Memory, as researchers have come to understand it, is distributed. It is not held in one single region, but spread across many different areas of the brain, each responsible for different pieces of the larger picture. The visual details of a memory, the color of a room, the shape of a face, may be processed and partly stored in regions related to vision. The emotional weight of a memory, the fear, the comfort, the joy, often involves structures deep within the brain associated with emotional processing. Even the physical sensations tied to a memory, like the feeling of rain on skin, or the sound of a familiar voice, are handled by separate, specialized regions. When you recall a single memory, say, a walk along a quiet shoreline years ago, you are not simply opening one file in one location. You are, in a sense, gathering fragments from many different parts of the brain at once, and weaving them back together into something that feels like a single, seamless moment. This is part of why memory can feel so fragile, and also part of why it can be so resilient. Because it isn't concentrated in one fragile point, it is spread wide, like light scattered across many surfaces. Now, to understand how a memory even begins, how it moves from a passing moment into something the brain can hold on to, we have to look at how memory unfolds over time. It often begins with something we call short-term memory, a brief, temporary space where new information first arrives. Short-term memory is remarkably limited. In most cases, it holds information for only a matter of seconds, or perhaps a few minutes, unless something happens to extend its life. If you hear a phone number and repeat it silently to yourself as you walk toward a piece of paper to write it down, you are engaging in something researchers call rehearsal. That repetition helps keep the information active, giving it a chance to be transferred somewhere more lasting. Without that rehearsal, without that repetition or attention, the information often simply slips away, not because anything went wrong, but because short-term memory was never designed to hold on to things for long. It is meant to be a temporary holding space, a kind of waiting room, rather than a permanent home. For information to move beyond that waiting room, to become something that can be recalled days, months, or even decades later, it must pass through a very specific structure deep within the brain. That structure is called the hippocampus. The hippocampus plays a central role in what scientists call consolidation, the process of converting short-term memories into long-term ones. It acts almost like a careful editor, or a quiet translator, taking fragile, temporary impressions and gradually helping to stabilize them into a more permanent form, often over the course of hours, or even while we sleep. This is part of why certain brain injuries, or conditions affecting the hippocampus, can create such striking gaps in memory. A person may be able to hold a conversation perfectly well in the moment, using their short-term memory just fine, yet be unable to form new lasting memories, because the pathway that would normally carry that information into long-term storage has been disrupted. The hippocampus, in a sense, is not where memories live permanently, but it is where many of them are born, shaped, and sent onward, into deeper, more lasting parts of the brain. But what does it actually mean, at the smallest level, for a memory to become long-lasting? What is physically happening inside the brain, at the level of individual cells? This brings us to something scientists call cellular consolidation. When you experience something meaningful, tiny connections between brain cells, called synapses, begin to change. These are the points where one neuron communicates with another, passing along small electrical and chemical signals. With enough activity, certain synaptic connections grow stronger, more efficient, more stable, and this physical strengthening is part of what allows a memory to become long-lasting. It's a beautifully strange idea, when you slow down and really consider it. A memory is not just an abstract idea floating somewhere in the mind. It has a physical form. It exists, in part, as an actual, structural change in the connections between cells. This is closely tied to something researchers often summarize with a simple phrase: cells that fire together, wire together. When certain neurons repeatedly activate together during an experience, the connections between them become reinforced. Over time, this repeated activation can carve something like a shallow path through a forest, faint at first, but increasingly clear each time it is walked. And this naturally leads to another important piece of the picture: the role that repetition plays in memory. Synaptic connections are not fixed once they're formed. They continue to respond to how often they are used. When information, or a skill, is repeated again and again, the related synaptic connections tend to strengthen further, making that memory more stable, more easily retrieved, more resistant to fading. But the reverse is also true. When connections go unused for long periods, they tend to weaken. This is often described using the phrase, "use it or lose it," a fitting way to describe how the brain quietly reallocates its resources over time. This is part of why a language learned fluently in childhood can become difficult to speak decades later without practice, even though it was once completely natural. It's not that the memory was destroyed. In many cases, remnants of it remain. But without ongoing use, the pathways supporting it gradually weakened, making retrieval more effortful, more uncertain. It also helps explain why skills practiced consistently, playing an instrument, solving certain kinds of problems, speaking in front of others, tend to feel almost automatic over time. Repetition doesn't just help you remember. It physically reinforces the very architecture that memory depends on. Given everything we've explored so far, it becomes clear that forgetting isn't caused by just one single thing. It happens through several different mechanisms, sometimes working alone, and sometimes overlapping. One of these mechanisms is simple decay over time, connections that go unused gradually weakening, as we just discussed. Another is interference, when new information competes with older information, sometimes making it harder to retrieve, or even partially overriding certain details. This is part of why, if you've ever moved to a new home, you might briefly find yourself reaching for the light switch in the location of your old one. The newer memory hasn't yet fully replaced the older pattern, and for a moment, the two compete. And a third major cause of forgetting relates not to what happens after a memory is formed, but to what happens in the very first moment it is created, a lack of attention during encoding. If your attention is divided, or distracted, at the moment something happens, the brain may never fully capture the details in the first place. This isn't a failure of memory. It's a failure of initial capture. You cannot forget something that was never truly recorded to begin with, which is why moments experienced while distracted so often leave only a faint, incomplete impression, if they leave any impression at all. This brings us to our final idea for tonight's exploration, one that touches something deeply human: the relationship between stress and memory. Stress doesn't simply make you feel overwhelmed in the moment, it can directly interfere with how memories are formed and recalled. When stress is high, it can disrupt attention, making it harder to properly encode new information. It can interfere with the delicate process of consolidation, making memories less stable. And it can even make retrieval more difficult, especially in moments of anxiety or emotional intensity, when the very thing you're trying to recall seems to slip further away the harder you reach for it. This is why people often describe forgetting simple, familiar information, a phone number, a name, a well-rehearsed line, during moments of extreme nervousness or fear. It isn't that the memory has vanished. It's that the stress response has temporarily disrupted the delicate systems responsible for finding it and bringing it forward. As we come to the end of this first exploration, a new picture of memory begins to take shape, quieter, and more intricate than the simple idea of fading candlelight we once imagined. Memory, it turns out, is not one single thing happening in one single place. It is a distributed, layered, constantly adjusting system, shaped by attention, by repetition, by emotion, by time, and by the quiet, ongoing work of structures like the hippocampus, translating fleeting moments into something more lasting. And forgetting, far from simply being a flaw in that system, often turns out to be part of its very design. In our next segment, we'll continue moving deeper into this landscape, exploring how different types of memory loss reveal themselves, how the brain changes with age, and what happens when this delicate balance begins to shift in unexpected ways. For now, let this first collection of ideas settle quietly in the background of your mind, needing nothing more from you than rest. Let's continue from where we left off, moving now into something even more delicate, the mechanics of forgetting itself, and how it shifts depending on the moment, the timescale, and the very structure of the brain's connections. We often think of forgetting as a single event. One moment, the memory is there. The next, it's gone. But research suggests something far more layered is happening beneath the surface, unfolding across different timescales, through different mechanisms, some lasting only seconds, others stretching across years. Let's begin with the smallest, most fleeting kind of forgetting, the kind that happens moment to moment. Have you ever been mid-sentence, only to suddenly lose the thread of what you were about to say? The thought was there just a second ago, fully formed, ready to be spoken, and then, without warning, it simply slips away. This isn't a sign of anything being wrong. It's a small, ordinary example of how short-term forgetting can be shaped by tiny shifts happening within the brain itself. Attention flickers constantly, even when we don't notice it. A sound outside a window, a passing thought, a slight change in how alert or tired we feel, these small fluctuations in neural activity can be enough to disrupt whatever was being held in that temporary, short-term space. The information wasn't lost in any permanent sense. The internal conditions simply shifted, just enough for it to slip through. It's a bit like trying to hold a reflection on the surface of water. If the water stays perfectly still, the reflection stays clear. But the smallest ripple, a breath of wind, a single drop, and the image begins to waver, blur, and disappear. The pond is not damaged. The water hasn't changed in any lasting way. The reflection simply needed stillness that, for a brief moment, wasn't there. This is one of the gentler forms of forgetting, temporary, shallow, and usually reversible, resolved simply by refocusing attention, or by encountering the information again. But not all forgetting stays at this shallow, momentary level. Some of it runs much deeper, unfolding not in seconds, but over weeks, months, or years, and this deeper kind of forgetting seems to involve something more structural, something happening within the very architecture of the brain's wiring. Researchers describe this as a kind of rewiring that happens around older memory traces. Over time, as new experiences accumulate, the brain doesn't simply leave old memories untouched in some static, frozen form. Instead, the surrounding neural pathways can shift and reorganize, and this reorganization can make it progressively more difficult to find the original path back to that older memory. Picture, for a moment, a well-worn trail through a forest, one that was walked often years ago, but has since been visited only rarely. As time passes, new trails form nearby, easier, more direct, more frequently traveled. Slowly, vegetation begins to grow across the older path, not because someone deliberately blocked it, but simply because it went unused while everything around it kept changing and growing. The old trail is still there in some form, beneath the growth, its outline persists. But finding it, and walking it again, becomes far more difficult than it once was. This is much of what happens, at a structural level, with certain long-term memories. It's not necessarily that the memory has been deleted. It's that the surrounding neural landscape has shifted enough that the original path has become obscured. To understand this more precisely, it helps to look at something scientists call an engram. An engram is the physical trace of a memory within the brain, the specific pattern of neurons and connections that together represent a particular experience. When a memory is formed, a distinct group of cells becomes associated with it, linked together through strengthened synaptic connections, forming something like a unique signature within the brain's vast network. Over time, these engram connections can change. Some connections may weaken. Others may become buried beneath newer, more active networks. And when this happens, the memory can become harder to access, not because the underlying trace has vanished, but because the specific pathway needed to reach it has been altered, weakened, or partially obscured by everything that has happened since. This is an important distinction, one that shifts the entire way scientists now think about long-term forgetting. It suggests that in many cases, what we experience as losing a memory may actually be less about the memory disappearing, and more about the accessibility of that memory quietly diminishing over time. The memory trace, the engram, may still exist, in some form, deep within the brain's structure. But the road leading to it may have grown fainter, more overgrown, harder to follow with each passing year. This idea has become central to one of the more compelling modern theories of forgetting. Rather than describing memory loss as erasure, as though information is being deleted, the way a file might be deleted from a computer, many researchers now describe it instead as a problem of accessibility. The information may still be there, encoded somewhere within the brain's architecture. What changes, over time, is how reachable it remains. This reframing carries a surprisingly hopeful undertone. If memory loss were purely about erasure, then once something was forgotten, it would, in a very real sense, be gone forever, unrecoverable, no matter what you did. But if forgetting is instead about accessibility, about pathways weakening rather than memories vanishing, then it raises a fascinating possibility. Under the right conditions, could some of those pathways be reopened? This question has led researchers into some genuinely remarkable areas of study, territory that, only a few decades ago, might have sounded closer to speculation than science. Some research suggests that certain memories, which appear completely lost, may still exist within the brain, but have become inaccessible due to disrupted signaling, rather than true erasure. In other words, the storage may still be intact. It's the retrieval signal, the pathway used to reach it, that is broken down. To explore this possibility more directly, scientists turn to a remarkable technique known as optogenetics. Optogenetics allows researchers, in controlled animal studies, to use light to activate very specific groups of neurons, cells that have been genetically modified to respond to particular wavelengths. Using this method, scientists have been able to identify the exact cells associated with a specific memory, the engram cells, and then artificially reactivate them, even in cases where the memory appeared, by every conventional measure, to be lost. And in a number of these studies, something extraordinary happened. When those specific engram cells were reactivated, the animals began to show behavioral signs that the memory, the seemingly forgotten memory, was still there, still intact, simply waiting for the right signal to bring it back into reach. It's a striking demonstration. It suggests that at least in some cases, what looks like forgetting from the outside may not reflect the true condition of the memory itself deep within the brain. The information didn't vanish. It became silent, disconnected from the usual pathways of retrieval, quietly waiting, perhaps for a signal that, under ordinary circumstances, might never come again. Interestingly, this phenomenon doesn't only appear under careful laboratory conditions involving light and genetics. Something similar seems to happen more naturally as well, often triggered by the everyday world around us. Some studies suggest that specific environmental cues, a particular scent, a familiar sound, a certain quality of light, a place once visited long ago, can spontaneously reactivate memories that seemed completely lost. In some cases, this reactivation doesn't just bring the memory back. It can also update it, blending old information with new context, reshaping it slightly as it re-enters conscious awareness. This offers a compelling explanation for something many people have experienced personally. Walking into a childhood home, years later, and suddenly recalling details that seemed entirely gone, the creak of a particular floorboard, the smell of a specific meal, a conversation from decades earlier, arriving with startling clarity. The memory wasn't erased all those years. It appears to have been dormant, waiting quietly for the right cue, a very specific key, fitting a very specific lock. This naturally raises an interesting, perhaps slightly unsettling question. If memories can be reactivated after appearing lost, does the simple act of remembering also change them? The answer, according to current research, is yes, and this insight has reshaped much of how scientists think about memory more broadly. Retrieval, the act of recalling a memory, is not a neutral process. It doesn't simply pull up a fixed, unchanging record, the way opening an old photograph might. Instead, each time a memory is retrieved, it can actually become temporarily unstable, entering a state where it is more open to influence. It can be strengthened by the act of recall, weakened if certain conditions disrupt it during that window, or in some cases, even subtly altered, shaped by whatever new context surrounds it in that particular moment. This is sometimes described using the term reconsolidation. When a memory is recalled, it briefly becomes malleable again, almost as if part of the original consolidation process is happening once more, before the memory settles back into long-term storage. And during that brief window, it can shift, ever so slightly, incorporating new emotional tone, new details, or new associations. This means that memory is far less like a fixed recording, and far more like a living, evolving impression, one that changes gently, almost imperceptibly, every time it is revisited. Each retelling, each recollection, has the potential to subtly reshape the memory itself. Taken together, all of this reveals something important about forgetting as a whole. It doesn't operate on just one single timescale or through just one single process. It unfolds across several distinct layers, each with its own rhythm, its own mechanism, its own depth. There is the fast, fleeting kind of forgetting, shifts in attention and brain state that cause information to slip away within moments, often resolved as quickly as they occurred. There is the intermediate kind, a slower rewiring of neural pathways surrounding older memories, gradually making certain memory traces more difficult, though not always impossible, to access. And there is the longer-term kind, a deeper reduction in accessibility, where connections weaken significantly over time, engrams become buried beneath newer patterns of activity, and memories drift further and further from easy reach, though, as we've seen, perhaps not always beyond reach entirely. 3 very different timescales, 3 very different mechanisms, yet all of them connected by a single, quietly reassuring thread. In many cases, forgetting may not represent true loss. It may represent distance. A signal grown faint. A path grown overgrown. A connection grown quiet, but not necessarily gone. As we move forward into the next part of this exploration, we'll continue to build on these ideas, moving deeper into how memory changes across a lifetime, and what happens when these delicate systems begin to shift in more noticeable ways. For now, let this second collection of ideas rest quietly alongside the first, layer upon layer, slowly forming a fuller picture of what it truly means to remember, and what it truly means to forget. Let's move forward now, carrying the thread of what we've already explored, and widening our view a little further. So far, we've looked closely at forgetting from the perspective of individual memories, how they form, how they weaken, how they occasionally return. But to truly understand forgetting, we need to step back, just slightly, and look at the brain not as a single mechanism, but as an enormous, layered system, with many different scales of activity happening all at once. We touched briefly on the idea that forgetting unfolds across different timescales, the fleeting, moment-to-moment slips caused by shifting attention, and the slower, deeper reconfiguration of memory traces that happens over weeks, months, and years. What's remarkable is that both of these processes are happening simultaneously, quietly overlapping, layered on top of one another like sheets of translucent glass. A brief change in brain state might cause a thought to slip away for a few seconds. And at the very same time, somewhere deeper within the brain's architecture, older memory traces may be undergoing a much slower, more gradual reshaping, the kind that takes years to fully unfold. These two processes don't operate separately. They exist together, constantly interacting, forming a kind of ongoing conversation between the immediate present and the accumulated past. And this brings us to something that may reshape how you think about memory even further. Forgetting doesn't only happen within individual brain cells, or within single connections between 2 neurons. It can also happen at a much larger scale, the level of entire networks. To understand this, it helps to imagine the brain not as a collection of isolated wires, each carrying its own separate signal, but as something closer to an enormous, interconnected web, where activity in one region constantly influences activity in many others. A single memory isn't held by one neuron acting alone. It emerges from the coordinated activity of many neurons, firing together in specific, organized patterns, across entire networks that can span multiple regions of the brain. When forgetting happens at this larger, network level, it isn't necessarily about individual connections weakening one by one. Instead, it can involve a shift in how entire groups of neurons communicate with each other, changes in timing, changes in synchronization, changes in the broader patterns of activity that allow a memory to be expressed as a coherent whole. It's a bit like an orchestra. A memory, in this sense, isn't the sound of one single instrument. It's the coordinated performance of many instruments together, each playing its part in time with the others. If the timing shifts, even slightly, if the violins drift out of sync with the rest of the ensemble, the overall performance can lose its coherence, even if every individual musician is still technically playing the correct notes. The memory, expressed as a network-level pattern, can become harder to recognize, harder to retrieve as a unified whole, even if pieces of it remain intact. This layered complexity is exactly why forgetting has become such a rich, multifaceted area of study within modern neuroscience. Today, researchers approach active forgetting from several different angles at once. At the cognitive level, scientists study how attention, distraction, and mental effort shape what we remember and what slips away. At the systems level, they examine how different brain regions, like the hippocampus and the cortex, interact and transfer information between each other over time. At the network level, they study how patterns of coordinated activity across many neurons give rise to memory, and how those patterns can shift or weaken. And at the cellular level, they investigate the molecular changes happening inside individual neurons and synapses, the tiny biological events that ultimately underlie everything happening at the larger scales above. Forgetting, then, isn't a single phenomenon with a single explanation. It's a multi-scale process, unfolding simultaneously across cognition, systems, networks, and cells, each level influencing and shaping the others in ways researchers are still working to fully understand. Given everything we've explored so far, it becomes tempting to view forgetting purely as a loss. Something going wrong. A system failing to hold on to what it should. But a compelling shift has occurred in how many researchers now think about this process, one that offers a very different way of understanding memory loss. Many scientists today describe forgetting not as a failure of memory, but as a necessary part of memory maintenance. Consider, for a moment, everything the brain encounters in a single day. Countless faces, sounds, conversations, sensations, decisions. If every single detail were retained with equal permanence and equal clarity, the mind would quickly become overwhelmed, cluttered with information that no longer serves any real purpose. Somewhere, the exact taste of a meal eaten years ago on an unremarkable evening. Somewhere else, the specific number of steps walked to your kitchen and back around midday, on an equally unremarkable afternoon. For memory to remain useful, the brain seems to require a way of clearing out what is no longer relevant, making space for what matters most in the present. This is why some researchers describe active forgetting as something the brain intentionally uses to remove memories that have become outdated or irrelevant to current circumstances. This isn't a flaw in the design. It may be an essential feature of it. This idea connects closely to something researchers describe as behavioral flexibility. Imagine, for a moment, if every strategy you had ever used to solve a problem remained equally strong and equally accessible in your mind, regardless of whether it's still applied to your current life. Old routes to work that no longer exist, competing just as strongly with the current path. Outdated methods for tasks, interfering with newer, more efficient approaches you've since learned. Without the ability to weaken or fade certain memories over time, adapting to new situations would become significantly more difficult. The old information would linger, cluttering the decision-making process, competing for attention alongside newer, more relevant knowledge. By allowing certain memories to fade, particularly ones tied to outdated patterns of behavior, the brain preserves its ability to remain flexible, to adjust smoothly as circumstances change, rather than remaining anchored indefinitely to patterns that may no longer serve any real purpose. This naturally connects to a broader idea, that forgetting may actually help cognition remain efficient. If every fact, every detail, every fleeting impression carried equal weight and equal permanence, focusing on what truly matters in any given moment would become extraordinarily difficult. Relevant information could easily become buried beneath an overwhelming accumulation of details that no longer hold any real significance. By allowing less relevant memories to gradually recede, the brain helps ensure that attention and cognitive resources remain available for what is currently important, rather than being divided endlessly among everything that has ever been experienced. In this light, memory decline doesn't have to be viewed purely as something harmful. In certain contexts, some degree of forgetting may actually help prevent cognitive overload, and in doing so, it may even make it easier to absorb and learn new information going forward. A mind that forgets selectively, rather than indiscriminately retaining everything, may in some ways be better equipped to keep learning, keep adapting, keep making room for what comes next. This idea extends even further, into something deeply personal and emotional. Forgetting may also play a protective role in mental health. Not every memory we form is a comfortable one. Difficult experiences, painful moments, distressing events, these, too, become encoded within the brain, just like any other memory. And in situations where such memories become intrusive, resurfacing again and again, uninvited, they can significantly affect emotional well-being. Some research suggests that the brain's capacity for forgetting may help reduce the frequency and intensity of these intrusive memories over time, gradually allowing their emotional weight to soften, even if the memory itself doesn't disappear completely. In this sense, forgetting isn't only about efficiency or flexibility. It may also serve as a kind of gentle, ongoing protection, a quiet mechanism helping the mind soften what might otherwise remain sharp and overwhelming indefinitely. To understand how any of this happens at the most fundamental level, we have to descend all the way down to the molecules themselves, the tiny biological components that ultimately make memory possible in the first place. Memory doesn't rely on permanent, unchanging structures within the brain. It relies on proteins, molecules, and cellular components that are constantly being built, used, and eventually broken down, replaced by newer versions through an ongoing process of natural turnover. This means that at a purely biological level, part of what allows memories to fade over time is simply the natural replacement of the molecular material that helps store them. The specific proteins involved in maintaining a particular synaptic connection don't last forever. They degrade and are gradually replaced, and through this ongoing process of renewal, some of the fine details underlying a memory trace can shift, weaken, or fade, purely as part of the body's normal biological turnover. Among the most important molecular players in this process are specialized proteins known as kinases and phosphatases. Protein kinases are molecules that help add small chemical tags, called phosphate groups, onto other proteins, a process that can strengthen and stabilize certain synaptic connections, helping to reinforce a memory trace. Protein phosphatases perform something closer to the opposite role. They help remove those same phosphate groups, a process that can weaken or destabilize synaptic connections, contributing to a memory becoming less accessible over time. Together, these two types of molecules function almost like a delicate biological balance, one reinforcing certain pathways, the other allowing others to soften, constantly adjusting the underlying structure of memory at the smallest possible scale. It's a striking thought, when you slow down and consider it fully. Somewhere, at this very moment, within the quiet architecture of your own mind, this exact balance is unfolding, microscopic decisions being made, again and again, about what should strengthen, and what should gently soften. As we bring this segment to a close, a broader, more compassionate understanding of forgetting continues to take shape. It is not simply a system failing under the weight of time. It is, in many respects, an active, adaptive, deeply necessary process, unfolding across cognition, across networks, across cells, and even down to the molecules themselves, quietly helping the mind stay open, flexible, and capable of continuing to grow. In the next part of our journey, we'll continue moving forward, exploring how these processes shift and change across different stages of life, and what happens when this delicate, adaptive balance begins to face new and unexpected challenges. For now, let this understanding settle quietly alongside everything we've already gathered, another layer, resting gently, as the fuller picture of memory continues, slowly, to come into view. Let's continue deeper into this landscape, moving now from the broad molecular balance we just explored, into something even more specific, the individual biological systems and structures that quietly shape whether a memory remains within reach, or slowly drifts beyond it. We spoke, a moment ago, about the ongoing turnover of molecular material within the brain, and about kinases and phosphatases gently tipping the balance between strengthening and weakening particular connections. But those molecular changes don't happen in isolation. They rely on an entire physical architecture, a kind of scaffolding, built from structural proteins and synaptic machinery, that determines whether a memory trace remains stable enough to be found again. These structural proteins act almost like the framework of a building. They hold synaptic connections in place, giving them shape and stability, allowing signals to pass efficiently between neurons. When this framework remains strong and well maintained, a memory tends to stay more accessible, more easily retrieved. But when this structural support weakens, whether through natural turnover, disuse, or the passage of time, the synaptic connections it once upheld can become less stable, and the memory they support can become correspondingly harder to reach. It's worth remembering that none of this happens in a fixed, unchanging way. This architecture is alive, constantly being maintained, repaired, and adjusted, much like a building that requires ongoing care to remain standing strong through the years. And just like a building, small shifts in that underlying structure can, over time, quietly determine whether something remains solid and reliable, or begins to shift and soften at the edges. For a long time, when scientists studied memory, most of the attention focused on how memories are formed. Which brain regions were involved, which chemical signals helped strengthen new connections, which processes allowed information to move from short-term into long-term storage. Far less attention was given to the opposite question: what chemical systems might be involved in actively helping memories fade? But that has changed significantly in recent years. Neurotransmitter systems, once studied almost exclusively for their role in creating and strengthening memories, are now understood to play an equally important role in forgetting. Neurotransmitters are the chemical messengers that allow neurons to communicate with one another, carrying signals across the tiny gaps between cells. And just as certain neurotransmitter activity can help stabilize and reinforce a memory, other neurotransmitter activity appears to actively contribute to weakening or suppressing memories that are no longer needed. One of the clearest examples of this involves a neurotransmitter called GABA. GABA is what's known as an inhibitory neurotransmitter, meaning its primary role is to reduce or dampen neural activity, rather than stimulate it. Within the hippocampus, that same structure we explored earlier, so central to forming new long-term memories, GABA signaling appears to also play a meaningful role in actively supporting forgetting. By reducing the activity of certain neurons, GABA can help limit or suppress the reinforcement of specific memory traces, gently quieting particular pathways that might otherwise remain overly active. In this way, the very same region responsible for helping memories take shape may also participate, through different chemical signals, in helping certain memories soften and recede. This dual role is a striking reminder of just how intricately balanced the brain truly is. The hippocampus isn't simply a memory-making machine, working in one direction only. It appears to be a place where both the strengthening and the softening of memory unfold side by side, shaped by different neurotransmitter systems working in coordination, sometimes at the very same time. To understand active forgetting more deeply, researchers have turned to animal studies, where the underlying biological pathways can be examined with far greater precision than would be possible in humans. And what they've found reveals a process that is remarkably intricate, involving several different systems working together in sequence. These studies have identified active forgetting pathways involving dopamine signaling, calcium signaling, and something called cytoskeletal remodeling, changes to the internal structural framework of the cell itself, the very scaffolding that helps maintain a neuron's shape and its synaptic connections. Dopamine, a neurotransmitter most commonly associated with reward and motivation, appears to play a surprising and somewhat unexpected role in forgetting as well. In certain proposed mechanisms, dopamine-driven signaling ultimately leads to the activation of two specific molecular components, one called RAC1 and another called cofulin. RAC1 and cofalin are both deeply involved in regulating the cytoskeleton, the internal structural framework we just mentioned, particularly within the small, delicate structures at synapses. When their activity increases, they can help trigger structural changes at the synapse itself, changes that can ultimately weaken or destabilize the specific connections involved in a particular memory trace. In this way, a signaling pathway that begins with dopamine can end with genuine physical remodeling within the neuron, quietly reshaping the very structure needed to retrieve a particular memory. Some of the clearest evidence for this pathway has come from studies involving fruit flies, a species long used in neuroscience because of how precisely their neural circuits can be studied and mapped. In these studies, active forgetting has been closely linked to specific dopamine neurons, and in particular, to something called dopamine nitric oxide cotransmission, a process where dopamine neurons release not only dopamine itself, but also nitric oxide, a small signaling molecule, at the very same time. Together, these two signals appear to work in coordination, influencing intracellular calcium signaling within neurons involved in memory, ultimately contributing to the active weakening of specific memory traces. Calcium, in this context, functions as an important internal messenger within the cell, helping translate external signals into meaningful internal changes. When calcium signaling shifts in particular ways, it can influence a wide range of downstream processes, including the very structural remodeling described a moment ago, gently steering certain synaptic connections toward weakening, rather than continued reinforcement. While much of this research has focused on simpler organisms like fruit flies, researchers have found that similar principles appear to apply within the more complex brains of mammals as well, including humans. In mammals, one particularly important mechanism involves something called AMPA receptor trafficking. AMPA receptors are specialized proteins located at synapses, playing a central role in allowing neurons to communicate effectively with one another. Their presence, and their precise positioning at a given synapse, directly influences how strong that particular connection remains. When more AMPA receptors are present and properly positioned, a synaptic connection tends to be stronger, more efficient, more capable of reliably transmitting signals. But when these receptors are removed, or relocated away from the synapse, a process researchers refer to as trafficking, the strength of that connection can decrease significantly. This trafficking process appears to be closely tied to memory instability and, in some cases, to active forgetting. As AMPA receptors are shifted away from particular synapses, the connections they once supported can weaken, making the associated memory trace correspondingly more difficult to retrieve. It's a precise, almost architectural process, receptors quietly relocating, and in doing so, gently reshaping the strength and accessibility of the memories those synapses once helped support. This same mechanism appears in more than one context. Changes in AMPA receptor trafficking don't only affect how strongly a memory is initially held. They also appear to directly influence how difficult that memory eventually becomes to retrieve, reinforcing just how deeply intertwined the physical structure of the synapse is with our ability to recall a specific moment from our past. Beyond neurons and their direct connections, there is another remarkable player involved in the forgetting process, one that isn't a neuron at all. These are cells known as microglia, the brain's resident immune cells, long understood to play a protective role, helping clear away damaged tissue and defend against infection. But in more recent years, researchers have discovered that microglia also play a direct, active role in shaping memory itself, particularly through a process involving the elimination of synapses. This process is known as complement-dependent synaptic pruning. The complement system is a group of proteins traditionally associated with immune defense, helping the body identify and eliminate harmful cells. Remarkably, this same system appears to also mark certain synapses for removal within the brain, tagging specific connections in a way that signals to microglia that they should be eliminated. Once tagged, microglia can then physically engulf and remove these marked synapses, effectively pruning away that particular connection entirely. This process has been directly linked to the forgetting of remote memories, memories formed further in the past, rather than more recent ones. Through this complement-dependent pruning, microglia appear capable of gradually eliminating the very synaptic connections that once supported a particular long-term memory, contributing to its fading, sometimes even its eventual disappearance, over extended periods of time. It's a striking image, when you consider it fully. Long after a memory was first formed, quietly reinforced through kinases, cytoskeletal remodeling, and receptor trafficking, an entirely different kind of cell, one belonging to the brain's immune system, rather than its network of neurons, may ultimately play a role in determining whether that memory's physical foundation continues to exist at all. As we bring this segment to a close, the picture of forgetting continues to grow richer and more intricate, layer upon layer. What began as a single, simple idea, that memories might fade with time, has revealed itself to be an extraordinarily coordinated biological process, involving neurotransmitters, structural proteins, intracellular signaling, receptor movement, and even the brain's own immune cells, all working together, across multiple systems, to shape which memories remain within reach, and which quietly begin to recede. In our next segment, we'll continue this journey, moving toward how these deep biological processes connect to real, lived experience, how memory and forgetting shift across the arc of a lifetime, and what happens as the brain continues to change with age. For now, let this intricate biological landscape settle quietly alongside everything we've already explored, each system, each signal, each quiet mechanism, another thread in the much larger, ever-unfolding tapestry of memory. Let's continue forward, carrying with us everything we've gathered so far, the neurons, the neurotransmitters, the immune cells quietly reshaping synaptic connections. Now we turn toward a broader question, one that brings several of these threads together. Not simply which cells are involved in forgetting, but what role forgetting seems to play in the very nature of memory itself, and how deeply woven it may be into the basic operating principles of the brain. We touched briefly, a moment ago, on microglia, and their role in pruning away certain synaptic connections. But microglia are only one member of a much larger family of cells known as glia, a broad category of non-neuronal cells that support, protect, and regulate the neurons around them. And it turns out, glial cells more broadly play a significant role in determining whether the specific synapses associated with a memory, the ones belonging to what researchers call engram cells, remain stable over time, or gradually weaken. Engram cells, as we discussed earlier, are the specific groups of neurons that come to represent a particular memory, linked together through strengthened synaptic connections formed at the moment that memory was created. Glial cells surround these engram synapses, quietly influencing their health and stability, sometimes providing support that helps preserve them, and at other times, contributing to processes that allow them to weaken or fade. In many ways, glial cells function almost like caretakers within this system, not creating memories themselves, but shaping the environment in which memories either continue to thrive, or slowly begin to recede. This brings us to an important and somewhat reassuring idea, one that echoes something we touched on earlier, but deserves to be explored a little further here. When a memory begins to fade, the brain doesn't necessarily destroy the entire memory outright. In many cases, what appears to happen instead is more selective, more precise. The brain may actively degrade, or even simply mask, specific synaptic connections within engram cells, rather than eliminating the memory as a complete, unified whole. This distinction matters a great deal. Degrading a memory trace suggests a partial weakening, certain connections becoming less effective, less stable, without necessarily disappearing altogether. Masking suggests something even gentler still, the memory trace remaining physically present, but somehow obscured, hidden beneath other, more dominant patterns of activity, making it far harder to access, without erasing its underlying structure. In either case, the evidence increasingly points toward the same conclusion. Complete erasure, in the truest sense of the word, may actually be far less common than we once assumed. Far more often, what we experience as forgetting may reflect memory traces that have been quietly degraded, or gently masked, altered in ways that make them harder to reach, rather than being wiped away entirely. One particularly fascinating area of research explores exactly when and how much of this degrading and masking seems to take place, and the answer may surprise you. It appears deeply connected to something we do every single night, often without giving it a second thought. Sleep. Some forgetting research suggests that sleep may play an active, selective role in shaping which memories remain strong, and which are allowed to weaken. Rather than treating every memory equally during sleep, the brain appears capable of distinguishing between memories that seem more valuable or relevant, and those that seem less useful, more incidental, or more likely to interfere with clearer, more important information. During sleep, particularly during certain stages associated with memory consolidation, the brain seems to engage in a kind of quiet curation, reinforcing certain memory traces, while allowing others, deemed less essential, to weaken or fade further into the background. This offers a compelling explanation for something almost universally experienced. Waking, after a full night's sleep, with certain memories from the previous day feeling notably clearer and more solid, while other, more minor details from that same day have already begun to blur, or vanish altogether. Sleep, in this light, is far from a passive state of simple rest. It appears to be an active period of memory reorganization, a nightly process during which the brain sorts through the accumulated experiences of the day, choosing, in some selective way, what deserves to be carried forward, and what can be gently released. There is another remarkable process connected to memory and forgetting, one rooted not in sleep, but in the ongoing growth of the brain itself, something researchers call neurogenesis. For much of the 20th century, it was widely believed that the adult brain could not produce new neurons, that the neurons you were born with, more or less, were the neurons you would carry for the rest of your life. But this turned out not to be entirely accurate. In certain regions of the brain, including, notably, the hippocampus, new neurons continue to be born throughout adulthood, integrating gradually into existing neural circuits. This ongoing production of new neurons, remarkable as it is, appears to carry an unexpected consequence. These newly born neurons, as they integrate into hippocampal circuits, can actually interfere with older, previously established memories, contributing to a distinct form of forgetting that researchers describe as neurogenesis-based forgetting. As new neurons settle into the existing network of the hippocampus, they don't simply add extra capacity alongside everything already present. They actively reshape the surrounding circuitry, adjusting how information flows, and how existing memories are accessed. This reorganization can make certain older memories, particularly ones tied closely to that specific circuit, more difficult to retrieve than they once were, not because those memories were directly damaged, but because the surrounding neural landscape has shifted around them, much like new construction gradually changing the layout of a once familiar neighborhood. This offers a particularly compelling explanation for why memory formation and memory loss often seem so closely intertwined throughout life. The very process that allows the hippocampus to continue learning, adapting, and forming brand new memories, also appears to be a process that can gently reshape, and sometimes obscure, memories formed earlier. Growth, in this sense, seems to exist in a delicate, ongoing balance with loss, not because one causes damage to the other, but because they are, in a very real sense, two expressions of the very same underlying system, continuously reshaping itself over time. All of this leads to a broader, perhaps deeper question. Beyond specific mechanisms like neurogenesis, sleep-based reorganization, or synaptic pruning, is there something even more fundamental at play, some underlying tendency within the brain itself that favors forgetting, unless actively counteracted? A growing body of research suggests that the answer may be yes. Some researchers propose that the brain relies on intrinsic signaling systems, internal biological processes that work continuously, quietly eroding memory traces over time, unless something actively intervenes to maintain and reinforce them. In this view, forgetting isn't simply something that occasionally happens, triggered only by specific external causes, like stress, distraction, or the passage of significant time. It may instead reflect an ongoing, built-in tendency within the brain itself, constantly working in the background, unless actively opposed by processes that reinforce and stabilize particular memories. This leads to a particularly striking proposal, one that quietly reshapes how we might think about memory as a whole. Some researchers have suggested that intrinsic forgetting may actually represent something close to the brain's default state, meaning that, without ongoing active maintenance, memories may naturally tend to fade over time, not because anything has gone wrong, but simply because that appears to be the underlying baseline tendency of the system itself. In this view, remembering isn't necessarily the effortless, automatic default we often assume it to be. It may actually require ongoing, active investment, repeated reinforcement, continued relevance, sufficient attention at the moment of encoding, and stable maintenance over time. Forgetting, by contrast, may require comparatively little, simply the natural passage of time, in the absence of that ongoing reinforcement. It's a humbling thought, in a way. It suggests that every memory you continue to hold on to, clearly and reliably, across the years, may represent not a passive accident of storage, but something closer to a quiet, ongoing achievement, the result of enough reinforcement, enough relevance, enough active maintenance, to resist what may be the brain's natural pull toward gradual fading. And this pull, interestingly, doesn't appear to operate independently of the world around you. Forgetting also seems to be shaped by the predictability of your environment. When a particular environment or set of circumstances remains fairly stable and predictable over time, certain memories tied to that environment may be maintained with relative consistency. But as contexts change, as the surrounding world becomes less predictable or shifts significantly from what it once was, this can influence which memories the brain considers worth preserving, and which it begins to treat as increasingly less relevant. The brain, in a very real sense, seems to calibrate its forgetting, at least in part, based on how much a particular memory continues to align with the world as it currently exists. Finally, it's worth pausing to recognize that active forgetting isn't a single, uniform process, driven by only one kind of trigger. Researchers describe it as taking multiple different forms, including both intentional and incidental forgetting. Intentional forgetting involves a more directed process, often connected to conscious motivation, deliberately trying to set aside a particular memory, perhaps because it has become distressing, unhelpful, or simply no longer useful in the present moment. Incidental forgetting, by contrast, happens without any such direct intention. It emerges instead as a natural byproduct of ordinary cognitive processes, memories fading not because the mind was specifically working to release them, but simply as part of the broader, ongoing maintenance occurring constantly within the brain, largely outside conscious awareness. As we bring this segment to a close, a fuller, more nuanced understanding of forgetting continues to take shape. It is neither a single failure, nor a single deliberate act. It is woven from many different threads at once, glial cells quietly shaping synaptic stability, sleep selectively curating the experiences of each passing day, new neurons reshaping old circuits simply by joining them, intrinsic signaling systems working steadily in the background, and the ever-shifting predictability of the world around us, all continuously influencing which memories remain close at hand, and which are gently, gradually, allowed to soften and fade. In our next segment, we'll continue moving forward, exploring how these deep biological patterns connect to real, everyday experience, how memory changes as we move through different stages of life, and what these processes can teach us about resilience, adaptation, and the quiet, ongoing nature of the mind itself. For now, let this fifth collection of ideas settle quietly alongside everything we've gathered so far, another layer, resting gently, as the fuller picture continues, slowly and steadily, to come into view. Let's continue now, moving into a part of this story that feels a little different from what we've explored so far. Until this point, much of what we've discussed has unfolded quietly, Automatically, beneath the surface of conscious awareness, molecules shifting, synapses weakening, glial cells and sleep cycles working in the background, largely without our direct involvement. But not all forgetting happens this way. Some of it, remarkably, can be deliberate. This is where we turn our attention to something researchers call intentional forgetting, the surprising capacity for a person to actively choose, in some meaningful sense, to set a particular memory aside. It might seem, at first, like an unusual idea. We tend to think of memory as something that simply happens to us, moments arriving, some staying, others fading, largely outside our control. But a growing body of research suggests that, at least to some degree, people are capable of deliberately suppressing unwanted memories, actively working to reduce their strength or their accessibility, rather than simply waiting for them to fade on their own over time. To understand how this is possible, we need to look toward a specific region of the brain known as the lateral prefrontal cortex. The prefrontal cortex, broadly speaking, is closely associated with higher-order cognitive functions, planning, decision-making, and various forms of conscious control over thought and behavior. The lateral portion of this region, in particular, appears to play a central role in intentional forgetting, functioning almost like a kind of oversight system, capable of exerting deliberate influence over other areas of the brain involved in memory. This points to something researchers describe as top-down inhibitory control. In simple terms, this means that certain higher-level regions of the brain, particularly within the prefrontal cortex, are capable of sending signals downward, toward other regions like the hippocampus, actively working to reduce or suppress activity there. Rather than memory suppression happening as some kind of passive drift, it can instead be actively steered, guided by intentional effort originating from these higher control regions. Researchers have identified at least two distinct strategies through which this kind of intentional forgetting appears to unfold, each relying on a slightly different underlying process. The first is known as direct suppression. Direct suppression works, in essence, by reducing activity within the hippocampus itself, specifically limiting its ability to retrieve a particular memory. Control signals, originating from the prefrontal cortex, travel toward the hippocampus, actively dampening the retrieval process for that specific piece of information. It's a bit like intentionally lowering the volume on a particular signal, rather than eliminating the source entirely. The memory trace may still exist within the hippocampus, but its ability to surface into conscious awareness becomes actively, deliberately restricted. The second strategy is quite different in approach, and is known as thought substitution. Rather than directly suppressing an unwanted memory, thought substitution works by deliberately replacing it with a different memory instead. Attention is redirected, intentionally, toward an alternative thought or recollection, one chosen specifically to occupy that same mental space. Over time, and with repeated practice, this substituted memory can become more readily accessible, while the original, unwanted memory becomes comparatively less prominent, gradually pushed further into the background, not through direct suppression, but through a kind of deliberate redirection. Both strategies, direct suppression and thought substitution, represent two distinct pathways toward the same broader outcome, intentional forgetting. But they appear to rely on somewhat different underlying brain processes to achieve that outcome, each engaging particular regions and particular patterns of neural activity in slightly different ways. This distinction matters because it suggests that forgetting isn't governed by a single, universal mechanism, even within the more deliberate, intentional category. Different strategies for forgetting appear to draw upon different underlying brain processes, each with its own particular signature, its own particular pathway through the brain's architecture. To study this phenomenon more directly, researchers have designed specific experimental approaches, allowing them to observe intentional forgetting as it unfolds in real time, within controlled laboratory conditions. These studies have provided compelling evidence that forgetting, at least in certain contexts, can be genuinely goal-directed and effortful, rather than simply a passive absence of remembering. In other words, choosing to forget something isn't necessarily an easier, lower-effort alternative to remembering. In many cases, it appears to require just as much active engagement, just as much genuine cognitive effort, as the act of deliberately trying to recall something clearly. One particularly revealing method used in this research involves what are known as directed forgetting experiments. In these studies, participants are typically presented with information, and then given explicit instructions, either to actively try to remember that information, or to deliberately try to forget it. By comparing brain activity across these two conditions, remembering versus intentionally forgetting, researchers have been able to observe clear, measurable differences in how the brain responds, depending on which particular goal a person is actively pursuing in that moment. These differences aren't subtle or ambiguous. They appear as distinct, identifiable patterns of neural activity, consistently different depending on whether someone is trying to hold on to a particular piece of information, or actively trying to release it. Perhaps the clearest, most consistent finding to emerge from this line of research involves a very specific pattern of brain activity, associated with successful intentional forgetting. When intentional forgetting proves effective, researchers have consistently observed two things happening together, in coordinated fashion. First, an increase in activity within the dorsolateral prefrontal cortex, a specific region closely associated with cognitive control and deliberate mental effort. And second, alongside that increase, a corresponding decrease in activity within the hippocampus, the very structure so central to forming and retrieving memories in the first place. This particular pattern, increased prefrontal activity paired with decreased hippocampal activity, offers a remarkably clear window into how intentional forgetting appears to unfold at a neural level. It suggests a kind of active, ongoing negotiation between two different brain regions, one region, the prefrontal cortex, exerting deliberate control, and another region, the hippocampus, having its natural retrieval processes actively dampened as a direct result of that control. It's worth sitting with this idea for a moment, because it reframes forgetting in a way that feels genuinely different from much of what we've explored so far. In earlier segments, we discussed forgetting largely as something that happens to memory, shaped by molecular turnover, synaptic pruning, sleep-based reorganization, and the brain's own intrinsic tendencies toward gradual fading. But here, we encounter something distinctly different. A form of forgetting that can be actively, deliberately pursued, not simply allowed to occur, but genuinely worked toward, engaging real cognitive effort and measurable neural resources in the process. This raises a subtle, thoughtful question, one worth carrying quietly forward as we continue this exploration. If forgetting can, in certain circumstances, be intentional, if the mind is capable of actively working to release a particular memory, rather than simply waiting for it to fade on its own, what does that suggest about the relationship between memory and personal agency? About the degree of influence, however partial, however effortful, that a person may hold over their own inner landscape of thought and recollection? These are not simple questions, and the science, even now, continues to explore them carefully, cautiously, without rushing toward easy answers. But what does seem increasingly clear is this: forgetting is not always something that merely happens to us. In certain circumstances, it can also be something we actively, deliberately do. A genuine act, requiring real effort, engaging specific brain regions, and producing measurable, observable changes in how the mind processes and releases particular memories. As we bring this segment to a close, we find ourselves standing at something of a turning point within this broader exploration. We have moved from the deep, often invisible biology of forgetting, molecules, synapses, glial cells, sleep, neurogenesis, towards something that feels, in certain moments, closer to genuine choice. A reminder that the mind, remarkable as it is never entirely passive in this process. Even here, even in the quiet act of letting something go, there is often more agency, more active participation, than we might initially assume. In our next segment, we'll continue building on this foundation, moving further into how these deliberate and involuntary processes intertwine across everyday life, and what they reveal about resilience, emotional regulation, and the intricate relationship between memory and well-being. For now, let this sixth collection of ideas settle quietly alongside everything we've already gathered, another layer, resting gently, as our understanding continues, slowly and steadily, to deepen. Let's continue moving forward, deeper into the fine detail of what happens, moment by moment, when the mind actively works to release an unwanted memory. In our last segment, we uncovered something remarkable: that intentional forgetting engages measurable, coordinated activity between the prefrontal cortex and the hippocampus. Now, we'll slow down even further, and look closely at the precise timing, the specific brain regions, and the surprisingly intricate sequence of events that unfolds within just a fraction of a second, whenever someone deliberately tries to let a memory go. Researchers studying this process have used direct brain recordings to observe what happens at the exact moment intentional forgetting takes place. And one of the clearest patterns to emerge involves something called alpha and beta oscillations. These oscillations refer to rhythmic patterns of electrical activity within the brain, occurring at particular frequencies. Alpha and beta waves, in particular, are often associated with a kind of active inhibition, a suppression of processing within certain brain regions. When researchers examine brain activity during intentional forgetting, they consistently observe stronger alpha and beta oscillations, compared to moments when someone is instead trying to remember something clearly. This finding offers a kind of rhythmic signature for forgetting itself, a distinctive wave pattern that seems to accompany the brain's active effort to suppress a particular memory. It's a striking thought, that beneath the surface of something as personal and quiet as choosing to let go of a memory, there exists a distinct, measurable rhythm, an electrical cadence that researchers can now observe and record. This leads to another compelling insight, one that gently reshapes how we might think about what actually happens to a memory during intentional forgetting. Some researchers have found that intentional forgetting may not necessarily destroy the memory trace itself. Instead, what appears to change are the top-down connections, the very same prefrontal pathways we discussed a moment ago, that would normally allow that memory to be retrieved. In other words, the memory may still exist, quietly, within the brain's architecture. What changes, through intentional suppression, is the pathway of access, the connection between control regions and the memory itself, rather than the memory's underlying existence. This echoes something we explored earlier, in a very different context, the idea that forgetting, so often, is not about erasure, but about accessibility. Here, we find that same principle reappearing, even within the realm of deliberate, effortful forgetting. This raises an intriguing question. If a memory isn't necessarily destroyed during suppression, what actually happens at the very beginning of this process? How does the brain first identify the memory it intends to suppress? The answer, gathered from careful experimental research, reveals something rather surprising. Before a memory can be effectively suppressed, it often needs to be, in some sense, partially activated first. This might seem counterintuitive at first. Why would the brain need to briefly activate a memory, in order to eventually suppress it? But when you consider it more closely, the logic becomes clear. In order to target a specific memory for suppression, the brain first needs some way of identifying it, of briefly recognizing its particular pattern, before inhibitory control can be directed toward it with any real precision. This suggests a two-stage process. First, a brief, partial reactivation of the unwanted memory. And only afterward, a wave of inhibitory control, working to weaken or suppress that same memory trace. This idea connects closely to what researchers describe as a brief window of reactivation, occurring just before inhibitory control fully takes over. When memory suppression is triggered, whether by an intrusive thought, an unwanted association, or a specific retrieval cue, the unwanted memory can briefly resurface, becoming momentarily active within conscious awareness, before the brain's control systems intervene to push it back down. This reactivation is typically brief, often lasting only a fraction of a second, but it appears to be a necessary part of the overall suppression process. This connects closely to something researchers have observed involving retrieval cues, specific external triggers, a word, an image, a particular sensation, that can briefly bring certain features of an unwanted memory back into partial awareness. When such a cue appears, it can momentarily reinstate certain elements of the unwanted memory, its emotional tone, a fragment of sensory detail, a fleeting impression, before inhibitory control mechanisms engage and work to suppress it once again. This offers a compelling explanation for something many people experience personally. An unwanted memory, seemingly successfully set aside, briefly resurfacing when triggered by an unexpected cue, only to fade back into the background again shortly afterward, gently pushed down once more by the same inhibitory processes. Given that this entire process unfolds so quickly, researchers have naturally wondered how the brain first recognizes that a particular memory is intrusive, and that active control is needed. This brings us to another key structure involved in this process, known as the anterior cingulate cortex. The anterior cingulate cortex appears to function almost like an early detection system within this broader network, responsible for identifying when an intrusive memory signal has emerged, and signaling, in turn, that inhibitory control may be required. Remarkably, researchers have found that this region can detect the need for control within roughly 500 milliseconds, half of a single second, of an intrusive memory beginning to surface. This speed is genuinely remarkable when you take a moment to consider it fully. Within less time than it takes to complete a single breath, the brain is capable of detecting an unwanted memory, recognizing its intrusive nature, and beginning to coordinate a response, all before the memory has any real opportunity to fully unfold within conscious awareness. Once this detection has occurred, the anterior cingulate cortex appears to help initiate a broader network of regions, working together to carry out the actual work of suppression. And this is where a particular region, one we touched on briefly in our previous segment, plays an especially prominent role: the right dorsolateral prefrontal cortex. This region appears especially important in suppressing episodic memory retrieval, the retrieval of specific, personally experienced events and experiences. When intentional forgetting is successfully carried out, this right-sided region shows increased activity, actively working to dampen the hippocampal processes that would otherwise allow that particular episodic memory to surface fully into awareness. Alongside this region, researchers have identified another important structure, known as the right inferior frontal gyrus. This particular region appears to play a more specialized role, helping to suppress the sensory components of an unwanted memory, specifically the vivid sensory details, the sounds, the images, the physical sensations that often accompany a particularly intrusive recollection. While the dorsolateral prefrontal cortex appears more broadly involved in suppressing the retrieval of the memory as a whole, the inferior frontal gyrus seems to focus more precisely on dampening its sensory vividness, its raw, immediate quality. Finally, there is one more region worth mentioning here, particularly relevant when the memory being suppressed carries significant emotional weight. This is the right medial frontal gyrus. This region appears to help regulate activity within both the hippocampus and the amygdala, a structure deeply involved in processing emotional responses, particularly during moments of emotional suppression. When someone is actively working to suppress a memory that carries strong emotional content, whether fear, sadness, or distress, this region appears to help coordinate a broader effort, working to calm both the memory-related activity within the hippocampus and the emotional intensity generated by the amygdala, together, in a more integrated fashion. Taken together, these findings reveal something genuinely remarkable when you step back and consider the full picture. Intentional forgetting isn't the work of a single brain region, acting alone. It involves an intricate, precisely coordinated network, unfolding within a matter of moments. The anterior cingulate cortex, detecting the intrusive signal almost instantly. The right dorsolateral prefrontal cortex, working to suppress the memory's retrieval. The right inferior frontal gyrus, dampening its vivid sensory details. And the right medial frontal gyrus, helping to calm the emotional intensity that so often accompanies our most difficult, most persistent memories. All of this, occurring within a matter of moments, largely outside conscious awareness, each region playing its own particular part within a much larger, remarkably coordinated system. As we bring this segment to a close, it becomes clear just how much complexity lies beneath something that, from the outside, can look deceptively simple: the quiet, private act of choosing to let a memory go. Far from being a passive process, or a single, isolated event, it involves a precise, rapid, deeply coordinated effort, unfolding across multiple brain regions, each contributing its own specialized role, working together within a fraction of a second, to gently guide a particular memory back into the background. In our next segment, we'll continue this exploration, moving further into how these deliberate processes interact with everyday emotional experience, and what they reveal about resilience, healing, and the mind's remarkable, ongoing capacity to adapt. For now, let this seventh collection of ideas settle quietly alongside everything we've already gathered, another layer, resting gently, as our understanding continues, slowly and steadily, to deepen. Let's continue now, building further on the delicate machinery we uncovered in our last exploration, that rapid, coordinated response involving the anterior cingulate cortex, the prefrontal regions, and the structures responsible for handling emotional intensity. Now, we turn our attention to something slightly different: how this entire process unfolds specifically when the memory involved carries real emotional weight, and what happens, over time, as suppression is practiced again and again. When researchers study the suppression of emotional memories specifically, they find that the process tends to unfold in two distinct phases, occurring one after the other, within a very brief span of time. The first is an early sensory phase. In this initial moment, the brain engages with the raw sensory qualities of the memory, its images, sounds, and physical sensations, much like the process we discussed in our previous segment, involving the right inferior frontal gyrus and its role in dampening vivid sensory detail. Only after this early sensory phase does the second stage begin, a later phase involving more deliberate emotional control. Here, the brain shifts its focus toward regulating the emotional intensity of the memory itself, working to soften its emotional charge, rather than simply its sensory vividness. This later phase tends to draw more heavily on regions like the medial frontal gyrus, working in coordination with the amygdala and hippocampus, gently calming the emotional response that the memory would otherwise continue to generate. This two-phase structure offers a subtle but meaningful insight. Suppressing an emotional memory isn't a single, uniform action. It unfolds gradually, moving first through the senses, and only afterward, through emotion itself, almost as though the mind approaches a difficult memory in careful stages, addressing what can be softened first, before turning toward what tends to be more deeply felt. Neuroimaging studies exploring this process consistently reveal a now familiar pattern, one we touched on in our previous segment, but worth revisiting here in this more emotional context. Successful suppression is reliably associated with an increase in prefrontal activity, alongside a corresponding decrease in hippocampal activity. This same coordinated pattern, control regions rising in activity, memory regions quieting in response, appears not only in more neutral, everyday forgetting, but also, quite consistently, when the memory involved carries genuine emotional significance. This raises a compelling question, one that researchers have worked carefully to answer. When suppression proves successful, what exactly is happening to the memory trace itself, deep within the brain's underlying structure? The evidence suggests that suppression-induced forgetting may directly affect the memory trace itself, rather than simply blocking momentary access to it. This is a subtly different idea from what we explored earlier, when discussing accessibility versus erasure. Here, some research suggests that repeated, deliberate suppression may not only make a memory harder to reach in a given moment, but may actually contribute to a more lasting weakening of the underlying trace, over time. This aligns closely with something else researchers have consistently observed. Suppressed memories, studied over extended periods, do appear to genuinely fade, becoming measurably less vivid, less easily recalled, as suppression continues to be practiced. This fading shows up clearly in how people describe their own memories following repeated suppression. Retrieval suppression, the deliberate effort to avoid recalling a particular memory, has been shown to reduce the vividness of that memory over time. Details that once felt sharp and clear can become softer, less defined, less immediate, the emotional and sensory clarity of the original experience gradually diminishing with each successful act of suppression. This reduction in vividness appears to have a clear neural counterpart as well, observable through neuroimaging. Suppression has been shown to reduce reactivation within a region known as the parahippocampal cortex, an area closely involved in processing contextual and spatial details associated with a memory, the surrounding scene, the physical setting, the broader context in which an experience originally took place. When suppression proves effective, this region shows notably reduced activity when the memory is later encountered again, suggesting that the richness of contextual detail normally associated with that memory has become measurably diminished, a quieter echo of what it once was. Beyond simply reducing vividness, memory suppression appears to carry meaningful consequences for behavior as well. Research suggests that suppression can directly reduce a memory's ongoing influence on later behavior, weakening not just how clearly a memory can be recalled, but also how strongly it continues to shape thoughts, emotional responses, and decisions moving forward. This finding carries real significance. It suggests that intentional forgetting isn't simply an internal, private experience, disconnected from everyday functioning. When practiced successfully, it can meaningfully influence how a person continues to move through the world, gradually loosening a difficult memory's grip on present-day thought and behavior, rather than leaving that influence untouched, quietly persisting in the background. Importantly, this effect appears to build over time, rather than occurring all at once. Researchers describe this as cumulative forgetting. Repeated suppression, practiced again and again, tends to produce a stronger, more lasting reduction in memory strength, compared to suppression attempted only once. This offers a hopeful, and perhaps intuitive, insight. A single attempt to set aside a difficult memory may only produce a modest, temporary effect. But with repeated practice, sustained gently over time, the underlying memory trace appears to weaken more substantially, each act of suppression contributing incrementally to an accumulating, longer-term change. Much of this research carries particularly meaningful implications within the context of trauma, and here, the findings become especially delicate and worth exploring with real care. Studies involving trauma-related memories have found that successful suppression is associated with reduced coupling between the brain's control regions and its memory-related regions. In simpler terms, this means that in successful suppression, the communication pathway between the prefrontal cortex and structures like the hippocampus and amygdala appears to weaken somewhat, allowing control regions to more effectively dampen activity within memory and emotion-related areas, without those regions continuing to strongly reactivate each other in return. However, this same research reveals something important and worth holding gently. People living with PTSD often experience significantly more difficulty engaging this suppression process effectively. For many individuals with PTSD, intrusive memories can resist the same kind of top-down control that tends to work more reliably in other circumstances. The coordinated network we explored earlier, less able to successfully dampen the intensity and recurrence of these particular memories. This isn't a reflection of personal failure or weakness. It reflects genuine, measurable differences in how certain neural systems function under the lasting effects of trauma, differences researchers continue to study carefully, in hopes of better understanding, and eventually supporting, more effective pathways toward relief. As we bring this segment to a close, we find ourselves holding a deeply layered understanding of memory suppression, one that moves gently between sensory detail and emotional depth, between single moments of effort and long, cumulative practice, between everyday forgetting and something far more delicate, the lasting echoes of difficult experience. Suppression, we've seen, is neither simple nor uniform. It unfolds in careful stages, shaped by specific brain regions, capable of genuinely softening a memory's vividness and its influence over time, though not always with equal ease for everyone. In our next segment, we'll continue this journey, moving toward broader questions of memory across the arc of a lifetime, and towards some of the final threads that bring this exploration closer to its completion. For now, let this eighth collection of ideas settle quietly alongside everything we've already gathered, another layer, resting gently, as our understanding continues, slowly and steadily, to deepen. Let's move forward now, carrying with us everything we've explored about suppression, its stages, its neural signatures, its deep connection to emotional regulation. In this next stretch of our journey, we'll widen the lens slightly, moving from the deliberate suppression of specific, often difficult memories, towards something broader, how memory control develops across a lifetime, and how the very act of remembering one thing can quietly shape our ability to remember something else. We've already seen how closely intentional forgetting is tied to emotional processing, the medial frontal gyrus working alongside the amygdala and hippocampus, softening both the sensory and emotional dimensions of a difficult memory. Taken together, memory control research reveals something worth pausing on. Suppression isn't simply a cognitive tool, separate from feeling. It appears to be deeply, fundamentally linked to emotion regulation itself, part of the same broader system the brain relies on to manage difficult feelings more generally, not a separate process existing off to the side. This connection helps explain why memory suppression so often appears in research alongside studies of emotional well-being, resilience, and coping. It isn't a coincidence that the same regions responsible for calming and intrusive memory are also central to regulating emotional responses more broadly. In many ways, learning to manage a difficult memory and learning to manage a difficult feeling may draw on much of the very same underlying neural architecture. Given how central this ability seems to be, it's worth asking a natural question. Is this capacity for suppression something a person is simply born with, fully formed? Or is it something that develops gradually, over time? Research suggests the latter. The ability to suppress unwanted memories appears to improve across childhood, growing stronger and more refined as a child develops. Young children, whose prefrontal cortex is still maturing, tend to show less capacity for this kind of deliberate control, while this ability becomes progressively more effective as the brain continues to develop through adolescence and into adulthood. This makes a great deal of intuitive sense when you consider everything we've discussed about the neural systems involved. The prefrontal cortex, so central to suppression, is among the last regions of the brain to fully mature, continuing its development well into a person's 20s. As this region strengthens and its connections to areas like the hippocampus become more refined, the capacity to deliberately, effectively manage unwanted memories appears to strengthen right alongside it. Memory control, in this sense, is not a fixed trait, but a skill that gradually develops, shaped by the brain's own unfolding maturation. There's another subtle but important detail worth sitting with here, one that quietly reframes how effortless this process might seem from the outside. The act of stopping retrieval, of actively preventing a memory from surfacing, appears to consume real biological energy. This might seem like a small detail, but it carries real weight. Suppression is not a passive absence of thought, requiring nothing from the brain. It is an active process, drawing on genuine metabolic resources, much like any other demanding cognitive task. This helps explain why sustained emotional suppression, practiced repeatedly over long periods, can sometimes leave a person feeling genuinely fatigued, not simply emotionally, but in a very real, physiological sense as well. Beyond this deliberate, effortful form of suppression, there exists another, quieter kind of forgetting, one that doesn't require any conscious intention at all. This is known as incidental forgetting, and it can occur simply as a natural side effect of retrieving a different memory altogether. This phenomenon is known more specifically as retrieval-induced forgetting. Here's how it tends to unfold. When you recall a particular memory, especially one that shares some connection or overlap with other related memories, the very act of retrieving that one memory can make competing, related memories less accessible, simply as a byproduct of that retrieval process, without any deliberate intention to forget anything at all. Imagine, for a moment, trying to recall the name of a particular restaurant you visited on a trip several years ago. As you search your memory, several possible names might briefly surface, competing with one another for your attention. Once you successfully identify the correct name, and successfully retrieve it, something interesting happens. The other names you briefly considered, the competing possibilities that didn't turn out to be correct, can become somewhat harder to recall in the future, weakened simply through their competition with the memory you ultimately retrieved. This is retrieval-induced forgetting in its clearest form. Recalling one memory doesn't leave neighboring, related memories untouched. It appears to actively suppress them, at least to some degree, as part of the very process of successfully retrieving the memory you were originally searching for. This form of forgetting, much like the intentional suppression we explored earlier, appears to rely heavily on the prefrontal cortex. When competing memories interfere with the retrieval of a specific, targeted memory, the prefrontal cortex appears to engage similar inhibitory mechanisms, helping to resolve that competition by actively dampening the competing alternatives, allowing the correct, targeted memory to be retrieved more clearly and more efficiently. Interestingly, this process carries a genuinely useful benefit, one that becomes especially clear with repeated practice. Repeated selective retrieval, deliberately recalling a specific piece of information again and again, tends to make future remembering more efficient over time. This offers a compelling explanation for why deliberate practice, testing yourself repeatedly on a particular piece of information rather than simply reviewing it passively, tends to be such an effective learning strategy. Each time a specific memory is successfully retrieved, competing alternatives become somewhat suppressed, while the targeted memory itself becomes increasingly reinforced, increasingly easy to access with each successful retrieval that follows. This effect has a clear signature within the brain itself as well, something researchers describe as cortical pattern suppression. When a particular memory is retrieved repeatedly, researchers have observed that the broader pattern of cortical activity associated with retrieving that memory becomes progressively more streamlined, more efficient, requiring somewhat less widespread neural engagement with each successful retrieval. In simpler terms, the brain appears to become better, more economical, at retrieving information it has successfully recalled before, gradually refining the process with continued practice. All of this connects to a broader, unifying theme, one that has quietly threaded its way through much of what we've explored in this segment and the last. Inhibitory control, the brain's capacity to actively suppress or dampen a particular process, isn't limited only to stopping unwanted physical actions, a reflex, an impulse, a movement begun and then interrupted. That same fundamental system also appears to extend into the realm of memory itself, helping to stop, or at least meaningfully dampen, the retrieval of particular memories, whether unwanted, emotionally difficult, or simply in competition with something else the mind is actively trying to recall. This shared foundation suggests something rather elegant about how the brain is organized. Rather than developing entirely separate systems for controlling behavior and controlling memory, the brain appears to draw on many of the same underlying inhibitory mechanisms for both, adapting a shared capacity for control toward multiple different purposes, depending on the particular demands of the moment. Researchers have proposed specific pathways underlying this connection, particularly those linking the prefrontal cortex with what is sometimes called the hippocampal complex, the broader network of interconnected structures surrounding and supporting the hippocampus itself. Through these pathways, the prefrontal cortex appears capable of exerting a kind of ongoing regulatory influence over memory retrieval more broadly, whether that influence is directed toward a single, specific unwanted memory, or toward managing competition among several related memories all vying for retrieval at once. As we bring this segment to a close, a clearer, more integrated picture continues to take shape. Memory control, we now understand, is not a single, isolated skill, reserved only for moments of deliberate, effortful suppression. It is a broader, more pervasive capacity, woven throughout the ordinary, everyday act of remembering itself, quietly shaping which memories rise easily to the surface, and which recede gently into the background, often without our conscious awareness that any such shaping is taking place at all. We are nearing the final stretch of this long exploration now, the last remaining threads still waiting to be gathered, before we bring this entire journey, gently, to its close. For now, let this ninth collection of ideas settle quietly alongside everything we've gathered so far, another layer, resting gently, as we prepare, together, for the final part of this exploration still to come. We arrive now at the final stretch of this long, quiet journey, one that began with a single, gentle question: why do we forget? And has since carried us through molecules, synapses, brain regions, and the delicate architecture of memory itself. Before we bring everything together, there remain a few last threads to explore, threads that move us from the deep laboratory science we've spent so much time within, towards something closer to everyday, lived experience. Until now, much of what we've discussed has involved the ordinary, healthy processes of forgetting, the quiet, adaptive mechanisms that shape memory even in a perfectly functioning brain. But forgetting can also take a very different form, one connected not to healthy maintenance, but to injury, illness, or disease. This is where we turn now, gently, toward the medical landscape of memory loss. Amnesia is perhaps the most widely recognized medical term associated with memory loss, and it can arise from a range of different causes. Head injury, stroke, brain tumors, and chronic alcoholism have all been linked to amnesia, each affecting the brain's memory systems through very different pathways, yet often arriving at a similar outcome: a significant disruption in a person's ability to remember. Within this broader category, researchers typically distinguish between two major types. Retrograde amnesia affects memories formed before the onset of injury or illness, disrupting access to the past. Anterograde amnesia, by contrast, impairs the ability to form new memories going forward, leaving old memories largely intact, while new experiences struggle to take lasting hold. These two forms of amnesia offer a striking illustration of just how many separate systems memory truly depends upon. A person experiencing anterograde amnesia might recall their childhood with wonderful clarity, and hold warm, detailed memories of people from years past, while being unable to remember a conversation that took place only moments earlier. It's a powerful, sometimes disorienting reminder that remembering the past, and forming new memories moving forward, rely on processes that, while connected, are not entirely one and the same. Beyond amnesia caused by injury or acute illness, memory loss can also emerge more gradually, through genetic and acquired brain disorders that unfold slowly over time. Among the most well-known of these is Alzheimer's disease, along with a broader category of related conditions known collectively as dementias. Unlike the sudden onset often associated with injury or stroke, these conditions tend to develop gradually, often over the course of many years, involving a slow accumulation of changes within the brain that progressively affect memory, and eventually, other cognitive functions as well. Fortunately, our ability to observe and understand this gradual progression has improved considerably in recent decades, largely thanks to advances in brain imaging technology. Tools such as MRI, magnetic resonance imaging, and PET, positron emission tomography, have given researchers a remarkable new window into the living brain, allowing them to track subtle structural and functional changes long before symptoms become clearly apparent. These imaging tools have proven especially valuable in tracking the progression toward Alzheimer's disease specifically, allowing researchers to observe early changes within the brain, sometimes years before significant memory symptoms begin to emerge. This growing ability to observe the brain's gradual transformation, quietly, patiently, over time, represents one of the more hopeful developments within this entire field, offering the possibility of earlier understanding and, ideally, earlier support. Even outside the context of specific disease, memory naturally shifts as we age, and researchers have found that this age-related change involves far more than the hippocampus alone. Age-related memory decline appears to be linked to broader structural changes across the brain, involving multiple regions and pathways working together, rather than any single area functioning in isolation. Interestingly, some of this age-related decline has been connected to a very specific molecular player, a gene known as RBAP48. Reduced activity of this particular gene has been associated with certain forms of age-related memory decline, offering researchers a specific, tangible molecular thread to study more closely, as they continue working to understand exactly how and why memory tends to shift as the years accumulate. Alongside the natural effects of aging, stress also appears to play a meaningful role in shaping memory over time. Both stress and aging have been shown to impair working memory, the kind of short-term, actively maintained memory we rely on for everyday tasks, holding a phone number in mind, following a multi-step instruction, keeping track of a conversation as it unfolds. But there is a genuinely hopeful detail worth holding on to here as well. Some animal studies suggest that certain aspects of this stress-related impairment may be reversible, at least under particular conditions. This hints at something important, that the effects of stress on memory are not necessarily fixed, permanent features of the aging brain, but may, in at least some cases, be responsive to change, to recovery, to intervention. This leads naturally to a broader, more encouraging idea, one worth carrying gently with you as we approach the end of this long exploration. While age-related memory decline is common, and in many ways a natural, expected part of the human lifespan, healthy habits and the brain's remarkable capacity for plasticity can meaningfully help preserve function over time. The brain, even well into later life, retains a genuine ability to adapt, to reorganize, to continue forming new connections in response to experience, learning, and care. This isn't a promise that memory will remain entirely unchanged throughout life. But it offers something quietly reassuring nonetheless, the understanding that the story of memory and aging is not simply one of steady, uncontrollable loss, but one that continues to involve real capacity for resilience, adaptation, and ongoing growth, even as the years continue to pass. And this brings us, gently, to where our long journey together now comes to rest. Across everything we've explored, from the earliest molecular details deep within a single synapse, to the broadest patterns of memory and aging across an entire lifetime, one theme has quietly returned again and again, appearing in different forms, across different scales, throughout this entire exploration. The brain spends an enormous amount of effort, constantly, quietly, carefully, balancing the act of remembering with the act of forgetting. And this balance reveals something important, something worth carrying with you long after this particular journey has ended. Memory loss, in so many of its forms, is not simply a failure. It is, remarkably often, an organized, deeply coordinated biological process, unfolding with real structure and real purpose, even when it appears, from the outside, as something going quietly wrong. We began this exploration with a simple question: why do we forget? And across everything we've discovered together, from cellular consolidation to engram accessibility, from GABA and dopamine to microglia and cortical pattern suppression, from the deliberate effort of intentional suppression to the natural drift of intrinsic decay, a much richer answer has slowly, patiently come into view. Forgetting is not the opposite of memory. It is not memory's enemy, working quietly against it. It is, instead, one of memory's essential tools, helping to keep the mind useful, adaptable, and efficient, clearing space where space is needed, softening what no longer serves us, and allowing what truly matters to remain close, clear, and accessible. The mind you carry, in this very moment, is not simply a record of everything that has ever happened to you. It is something far more alive than that, a living, ongoing collaboration between remembering and releasing, between holding on and gently letting go, continuously shaped, continuously refined, moment by moment, across every day of your life. There is something quietly comforting in this understanding, if you allow yourself to sit with it fully. Forgetting is not evidence of a mind failing. Far more often, it is evidence of a mind working exactly as it was designed to, tending carefully to itself, making room, again and again, for whatever comes next. As this long exploration finally comes to its close, there is nothing more you need to hold on to, nothing more you need to remember, no effort required of you at all. You can simply let these ideas settle now, quietly, gently, the way memory itself so often does, drifting slowly into the soft, patient background of rest.